following study looks at the ventral tegmental area and how astrocytes orchestrate avoidance and approach behavior. The ventral tegmental area, also known as the VTA, has been known to mediate learning of approach and avoidance through dopaminergic neurons. Now, it is known that dopaminergic neurons are responsible for reward behavior. So essentially, if an animal sees something and it creates benefit for that animal, the dopamine will tell that animal that this is good and award reward behavior. However, it is a little bit more difficult to understand how, when an animal sees a behavior and it creates a negative effect, how that neurocircuitry works. One thing to understand about neurocircuitry is the most common neurotransmitters used is GABA and glutamate. Now, GABA will yield inhibitory responses, whereas glutamate will result in excitatory neurotransmitters in the central nervous system. And with GABA, you will yield iPSCs, known as inhibitory postsynaptic currents, whereas glutamate will yield EPSCs, known as excitatory postsynaptic currents. Also known is that astrocytes are known to manage uptake and destruction of various neurotransmitters in the synapses as glial cells. And in this study, the main enzyme looked at is GLT-1, which regulates the uptake of glutamate. Another aspect of the study is that the study uses optogenetics, which is a method of using light to activate or stimulate astrocytes or neurons. And in this case, channel rhodopsin 2, also known as CHR2, was used, which is a genetic mutation that allows the stimulation of neurons and astrocytes from light waves. First, the study looked at how astrocytes work to activate GABAergic neurons. Now, when channel rhodopsin 2 was used to stimulate these astrocytes, GABAergic neurons were also stimulated. This was found to be due to GLT-1, which we will look further into in later aspects of the study. Now, astrocytes will reduce the amount of GLT-1 to increase activation of GABAergic neurons. In other words, if you reduce the amount of destruction and uptake of glutamate, these GABAergic neurons will become stimulated. This study did not look at postsynaptic glutamergic neurons. Another aspect of the study was how astrocytes work to inhibit dopaminergic neurons. Now, again, using channel rhodopsin 2 to stimulate these astrocytes, recordings from the VTA dopaminergic neurons were taken, and it was found that inhibitory responses were recorded. Now, if you block the GLT-1 in these astrocytes, and you can see this in figure I, these dopaminergic neurons would express EPSCs, excitatory postsynaptic currents, as opposed to iPSCs, which means that GLT-1 is responsible for inhibiting these dopaminergic neurons in the VTA, meaning that these reward systems were blocked, and an increase in GLT-1 synapses, or rather a decrease in GLT-1 would yield iPSCs, and an increase would yield EPSCs. And in order, just as a visual representation, in the top right hand of the corner, you can see how channel rhodopsin 2 was expressed in astrocytes and not in the neuronal soma. Another visual representation is an example of how the microcircuitry in the VTA. So you can see how there are astrocytes, the GABAergic neurons, which are represented by circles, and then the dopaminergic neurons represented by hexagons. How these three different two neurons and astrocyte were activated, as well as the neurons activated by channel rhodopsin 2. Next, the study worked to understand and prove, rather, that GABAergic neurons were inhibiting these dopaminergic neurons. So, when GABAergic neurons were blocked by succulene, IPSCs were no longer seen from dopaminergic neurons, which you can see from figure B, as when these GABAergic neurons were firing, you would see inhibitory responses upon the dopaminergic neurons, whereas if the GABAergic neurons were not firing, you would not see these iPSCs. Next, the study looked at how inhibition of the astrocytes would yield specific behaviors. So the study had proven that GABAergic neurons were acting upon the dopaminergic neurons. So astrocytes had been found to act upon both the GABAergic and dopaminergic neurons, and the GABAergic neurons would act upon the dopaminergic neurons. So now the study looked at to see how specific behaviors that could be visualized would happen. So when the astrocytes with channel rhodopsin 2 were activated, they would yield avoidance behavior, and it can be seen from figure B. When the laser was on, the animals would stick to the sides away from the center, which means that they would avoid open areas. When the laser was off, you can see how they would approach the middle of the area, and then when the laser was turned back on, they would then approach more so of that middle area. And astrocytes that did not have GLT, or did not have the channel rhodopsin where those astrocytes could be activated, did not express this behavior if the laser was turned on. Furthermore, if you were to inhibit GLT1 in neurons, whether they had channel rhodopsin 2 or not, you would see that avoidance behavior was not seen or visualized from this test. Some of the conclusions and applications of the study is, the main conclusion, obviously, is that astrocytes, and more specifically GLT1 from astrocytes, regulate avoidance behavior, which can be seen from the figure on the bottom right hand of the screen. And some possible applications from this study is that patients who suffer from anxiety are prone to increased avoidance. Therefore, if you knock out the GLT1 enzymes in the VTA, GABAergic neurons could possibly reduce the anxiety. And in other words, if you reduce the amount of glutamate that is being destroyed, you'll have more of an approach behavior rather than an avoidance behavior, because the dopaminergic neurons would be stimulated. On the other hand, in patients that are suffering from drug overdoses, such as cocaine, which causes the blockage of dopamine intake seen from the bottom right hand of the screen, a increased expression or stimulation of GLT1 and astrocytes in the VTA could inhibit dopaminergic neurons, meaning if you are having these dopaminergic neurons releasing an increased amount of dopamine, you could increase the amount of astrocytic activity, which increases the amount of GLT1 activity and inhibits these dopaminergic neurons. Now, one thing, however, unfortunately, that this study did not go into is further applications of how this article can be used in clinical researches, and these the clinical studies from this article are more so of a assumed possibility. However, the anxiety aspect of this article was partially discussed in the introduction. The other confusing aspect is GLT1 an increased amount of GLT1 will reduce the amount of EPSCs on dopamine, but will also reduce the amount of dopamine seen in GABAergic neurons. And it's unclear as to how they would inhibit GLT1 in the VTA and not have both the GABAergic and dopaminergic neurons express the same amount of levels. So it's the study was a little confusing as to how these the knockout of GLT1 really yields these results. However, the importance of this study looks at how astrocytes and glial cells are actually important for synapses, which in previous years and studies, these glial cells have been found to not be, or were thought to not have much importance. However, the study kind of proves otherwise.